Hey, hello, I'm Jaden. To my left is Aaron, and to my right is Kyle. And today we'll be talking about biotoxicity and how we use silver nanoparticles to basically kill microorganisms. Okay, so I first wanted to start with how we made silver nanoparticles in our lab. So we used silver nitrate, and then we reduced it, which was basically means added electrons to silver nitrate using sodium citrate, which I think another group talked about. And then we put that on a hot plate and used excess sodium citrate to prevent the particles from combining together to create something larger than a nanoparticle, because nanoparticles have the right amount of surface area and like structure so that we can easily kill bacteria. And then after that, we basically used heat to make sure the nanoparticle was the right size. Okay, so a few applications of silver nanoparticles would be anything that basically kills microorganisms, such as I think they are used in bandages and also like mouthwash. So yeah, it's pretty interesting.
So in this experiment, we tested the effectiveness of 4 different ways of, or at least attempts to kill cells. So we used yeast cells, and pretty much we determined whether or not they were killed by using a manometer to measure how much carbon dioxide was being produced. So obviously, if there are cells due to cellular respiration, more carbon dioxide will be produced. But if whatever we put in killed the cells, then less carbon dioxide would be produced. So for the analogy, the hearts represent the nanoparticles. So the white heart is the silver nanoparticles, and the darker hearts are the gold nanoparticles. So pretty much the first part of the analogy is how they enter the ride. So if you'll see, both of them have to have like free hugs to get onto the ride. So I think this is demonstrating how nanoparticles enter through endocytosis, which is pretty much the membrane allows them to come in and then sort of hugs them and brings them into the cell. So that's how the nanoparticles get in, because they have no charge, but the ions, which are the triangles, because they do have a charge, they can't get in through endocytosis, so they have to enter through ion channels. So as you can see with the triangles, there's a little tunnel. So to get on the ride, they have to enter through the ion channels. And then the bulk silver, which is the circle with triangles inside. So as talked about by the earlier group, the bulk silver can't fit in, so the ions have to come off of the bulk silver to get in, which is much slower. So as you can see, I think the speech bubble says like must be this small to ride. So pretty much they have to separate. The triangles have to come off of the bulk to go on the ride. And as you can see, there's only 2 triangles on the ride, while all the other rides are filled up because it's such a slow process. So after the entry, the next part of the analogy is the damage that they all do to the cell. So as you can see, the silver nanoparticles on the left, which are the white hearts, there's a speech bubble that says this ride is broken. So pretty much silver nanoparticles do the most damage, and therefore, as you can see, they pretty much the ride fails, which pretty much means the cell would have died in the experiment, and we saw that as there was less carbon dioxide being produced. But as you can see on the right side, the gold nanoparticles, it still says this ride is so fast. So actually, since gold, it doesn't have the toxicity of silver in terms of killing the cells, so it wouldn't be effective if you insert it into the yeast cells to kill them. So that ride is still fully functional, and as you can see, the one with the bulk silver is also fully functional because since the silver ions enter so slowly, it doesn't have enough of a harmful effect on the cells. And then just the pure silver ions, they're kind of in between, where they had a negative effect on the cells as the carbon dioxide went down, but not as much as the nanoparticles. So it just says this ride is slow. So it's kind of like not completely killing them, but it's not killing all of them, but killing a large portion of them.
So scientific analogies have limitations, and it's extremely difficult to encompass many concepts in one analogy. And as a result, our analogy doesn't quite encompass every concept we learned in the process. So one thing that the drawing leaves out is the process in which our silver products actually kill the bacteria or the target cell. So after each silver product ultimately sheds off silver cations, the cations then oxidize the oxygen atoms present in the cell, and that ionizes and makes the oxygens extremely reactive. And these new reactive oxygens then bind on to the nonpolar hydrophobic components of the cell's membranes, which includes the mitochondria membrane and the actual cell membrane. And it turns these nonpolar components polar and hydrophilic. And without any hydrophobic components to the cell membranes, the cell membrane is unable to conserve its phospholipid bilayer structure, and ultimately it just collapses into itself. And this obviously kills the cell, and yeah, that's basically how it kills bacteria or unwanted cells.
Any questions?
Okay, I'll start. I have a couple. So first, so you mentioned how the silver is toxic to the yeast and the gold is not. So they're both nanoparticles. So why is one toxic and one not toxic? What's the difference?
So silver has a charge of +1, whereas gold has a charge of +3, and so silver is more reactive or more easily oxidizes the oxygen than the gold.
Right, so if you see like silver, just like in general, like if you have silver plates or silver spoons or silver jewelry or whatever it is, what happens to silver over time normally?
It reacts.
It reacts to,
Like everything surrounding it because it's so reactive because it's +1.
it oxidizes, right? It
Yeah.
tarnishes. So gold, does it do the same thing?
No.
Right, so is it reactive to gold?
No.
Not really, right? Good. Another question I had is, so these ones, I do see that they are trying to capture differences in size with the triangles and the circles having more triangles and the hearts having less triangles. What is the actual size ratios of the three types of silver?
So the silver ions are actually the smallest, and then the nanoparticles are actually bigger than the silver ions. But because they have no charge, they can enter through endocytosis, while the silver ions can't. And then the bulk silver is the biggest.
Right. So in terms of like the actual size, though, like how big is an ion? How big is the nanoparticle? And then roughly, how big is the bulk?
Well, an ion would probably be like picometers in size, probably, right?
It's it's like fractions of a nanometer.
Yeah, and well, obviously the nanoparticles are around in the nanometer range. So like probably maybe 5 to 20 nanometers, and then bulk is like probably micrometers.
Yeah. So really, really big differences in size. Oh, so many questions now. Okay, cool. Here, let's start. Go ahead.
This question goes back to what was brought up earlier, where someone consumed so much silver that he turned blue. And it seems to be not hurting him, but then my thought was, will he stay blue? Or will the body expel the silver?
We didn't. I'm not 100% sure about this, but I assume that the body would continue to make more and more cells, and therefore it wouldn't. It would eventually go back, but I can't.
And I also think some materials can't be dispelled from the body really easily, and I'm pretty sure silver is one of them.
Yeah.
So time will tell, but maybe not, he'll stay
Yeah.
blue the rest of his life.
As the silver oxidizes, right? Does the silver ever lose its potency or the ability to kill the yeast through time? And if it oxidizes, can you polish it again or get it straight back?
I think when the silver oxidizes the oxygen, it just kind of loses its, or gains the electron. And I think it just, yeah, I don't think it can.
I think in theory it is possible to reverse it, but in practice, I'm not sure if it would be possible.
And then was there one more question? Yes?
Can you explain how you quantified the damage done in each sample?
So we pretty much had the manometer capped onto the combination of yeast, sugar, and water with a stopper. And pretty much if the yeast was creating carbon dioxide, the manometer, the water would move. So you could see that there's more gas, and therefore we measured how much the water moved up, and that meant that there's more carbon dioxide being produced. While for certain ones like the nanoparticles, the water would actually move down because it was using oxygen for cellular respiration, but then eventually the yeast cells were dying, so it didn't complete cellular respiration and not enough carbon dioxide was produced, so the water level would go down. So we just measured how much it went up or down.
Any last questions? Oh. Wait, sorry, Elaine's gonna, or you're gonna get there you go.
So when you synthesize the nanoparticles, you created 10 nanometer nanoparticles. How did you synthesize them and know how to grow them to a specific size?
I think eventually just the sodium citrate decomposed completely, so they wouldn't expand more. But yeah.
Was there something to do with plasma resonance?
I'm not sure, but maybe some of my teammates can respond.
So here, let me help you guys out. So you guys learned from plasmonics that what happens if you're observing a color change.
Oh, the particle might be getting bigger, or like might have like more arms, more surface area.
So then you're using what to determine what then when you were synthesizing your silver
Oh, we were
nanoparticles?
using light to determine, or the color
The color, yeah.
to determine. Oh, okay.
So when it turned more yellow, that's when we took it out.
Any last questions? All right, thank you guys.
Thank you.